Buenas tardes. Sean bienvenidos al Seminario Departamental del Laboratorio de Biotecnología Ambiental, que esta semana nos comparte el tema Análisis in silico de enzimas bacterianas asociadas a la degradación del polietileno. La ponente será la maestra en ciencias Aminat Olamide Shokunbi, quien es estudiante del doctorado de este centro y participa en el Laboratorio de Biotecnología Ambiental. Ella cuenta con una licenciatura en zoología por la Universidad Federal de Agricultura de Abeokuta, Nigeria, y una maestría en parasitología y bioinformática por la Universidad de Lagos, Nigeria. Ha recibido premios en sus trabajos sobre malaria, enfermedades tropicales desatendidas y enfermedades infecciosas. Actualmente su investigación se centra en enzimas bacterianas capaces de degradar plásticos resistentes como el polietileno y combina búsquedas en base de datos y herramientas bioinformáticas para comprender cómo funcionan estos microorganismos e identificar la base enzimática y molecular de la degradación de plásticos. Maestra, bienvenida.
Buenas tardes a todos. Mi nombre es Aminat Olamide Shokunbi. I'm here to give you a presentation on my thesis advance titled In Silico Analysis of Bacteria Enzymes Associated with Plastic Degradation. As we all know, the versatile properties of plastics, such as their durability, has increased their demand throughout agricultural industry, the consumer goods industry, construction industries, and many more. So because there's increase in demand, it has led to the increase in the productions of these plastics. So annually, scientists have found that over 350 to 400 million plastics are produced in a year. And of these 400 million plastics, 5 to 13 million of them enters into the ocean, negatively affecting the ecological environment. By 2050, scientists fear a pandemic of plastic accumulation because already we do not have a solution to plastic accumulation now. And of this, like I earlier mentioned, I said that because of the increasing demand of these plastics, over 400 million of them are produced annually. And of these 400 million plastics, polyethylene accounts for 38% of them. That is why we are interested in polyethylene, because it is the most produced in all of them. And why is polyethylene the most produced? Because polyethylene is made of ethylene ethylene monomers. We all know that ethylene is the most abundant petrochemical in the world. They are easily available and are extremely cheap. That's why they are the most produced. Now, polyethylene is a long-chain hydrocarbon. They are made of strong covalent carbon-carbon and carbon-hydrogen bond, and this is the reason why they are stable. This covalent bond are the reason why they are stable and their resistance to degradation. They do not react with acid, base, or water. That's why it's easier for them to be used in the packaging industry, because when you put your food inside in a plastic and it's extremely safe, it does not react with either the component of the plastics. It made them extremely safe. As a result, they are used worldwide. The three different types of polyethylene. We have the high-density polyethylene, the low-density polyethylene or branch low-density polyethylene, and the linear low-density polyethylene. Now, the higher the branches, the weaker the intermolecular forces and the lesser the tensile strength between them. That's why as difficult, as recalcitrant as polyethylene is, high-density polyethylene is more recalcitrant because they are bonded together by van der Waals force, and it takes a lot to degrade them. Now, as we know that the problem of plastic accumulation is enormous, it's not like scientists have not tried. They've tried a lot. They try to use traditional methods like burning them, and you know that when you burn plastics, they affect the ecosystem. They deplete the ozone layer and causes greenhouse effect. So it's important that we look for an eco-friendly means. And that's why we think about microorganisms, because we need an economically friendly means, a means that does not affect human or animals. And then we think about microorganisms. Now, microorganisms are not just they are not just environmentally friendly. They are equally economically friendly. So it takes just a little to do more. Entonces, we decided to use microorganisms. Not just us, but of course, scientists decided to use microorganisms because they're extremely cheap and they're environmentally friendly. Now, to demonstrate the feasibility of our studies, we showcase successful researches that have been carried out. Just recently, this year, in 2025, this year that we have, Kim et al. Discovered that Pseudomonas produce alkane monoxygenases and they're able to degrade polyethylene. A lot of them like that. We also discovered in 2023 by Tao that lacases are able to degrade polyethylene, lacases from Rhodococcus rubber. Also, Zhang et al. Zhang has done a lot of researches with polyethylene, and then he found out that a consortium of bacteria, Anoribacillus and Bacillus, are able to degrade polyethylene. Also, Pramila in 2015, they also found out that Acinetobacter has extracellular lacase that could degrade polyethylene. In our studies, we found out that enzymes activities, the rate of degradation by enzymes activities affect the rate of degradation. It affects it so that, I'm going to use the example in Zhang et al. Zhang found out that a molecule of lacase from Cyclobacter could catalyze approximately 42 reactions per second. What does this mean? When we talk about enzyme degradation, the catalytic constant is very important. That is the turnover number of an enzyme. The rate at which an enzyme is able to convert a substrate to a product is very important in enzyme studies. How fast do they do it? Does it take time? How can we reduce this time if it takes time? So in enzyme studies, it is important to also take note of the catalytic constant. Now we go to the justification of our studies. Why did we decide to do this study? We know it is no more news that plastic accumulation has become a problem. A study in New York in 2020 discovered that plastics are found in the fecal sample of a child. That's a problem. If we've already found plastic in the fecal sample of a newborn child, of course it's not newborn, maybe a month, that's a problem. So we cannot wait till the time that this plastic accumulation become a problem before we start acting. We decided to start acting now. And how do we decide to act? We decided to use eco-friendly means, which are microorganisms. Now, in our study, we did not just decide to use microorganisms, we decided to use in silico analysis. Why? Because in silico analysis enables a large-scale prediction of enzymes candidate based on sequence homologue, concept motif, structural models, and evolutionary relationship. This will ensure the discovery of both known and unknown and new enzymes, So we decided to use in silico analysis because, of course, scientists believe that organisms that share similar homologue, there's a probability that they do almost the same thing because sequence homologue has not to do with just physical, but traced down to evolutionary. Now, one of the most important parts of in silico analysis is concept motif. Because concept motifs serve as biochemical fingerprint of enzymes activities, we decided to, in silico analysis, deal more with concept motifs. Because enzyme functions are ultimately defined by concept patterns. Now, even in a case where there's mutation, nothing affects concept motif, because like I said, they are the fingerprint of enzyme activities. Just like we know that they are the active site residue where the catalysis actually happen. So we concentrate more on the concept motif. Also, we also checked structural, we found that structural modeling and molecular docking predict enzyme substrate interaction. Of course, it's when we know the catalytic residue and we're able to check the binding residue as well. It will help us know how these enzymes carry out this function, and it will give us insight into a lot of things. Now, this approach accelerates enzyme discovery and reduces laboratory workload. if we are able to identify new bacterial enzymes, it will reduce the problem of plastic accumulation in the environment. As a result, this research is not just timely, but is also necessary. The hypothesis of our study, we envisage that genomic analysis and functional prediction based on conserved protein domains and sequence motifs allow the discovery of new bacterial enzymes capable of degrading polyethylene. General objective, we envisage that we'll be able to identify, characterize, we'll be able to identify and characterize novel bacterial enzymes associated with polyethylene degradation through the analysis of their genome and enzymatic profile using computational and comparative bioinformatic tools. Our general objective is to compare and curate bacterial species reported to degrade polyethylene through a systematic review of databases and literature. Our second specific objective is to identify, annotate, and compare selected bacterial enzymes that share sequence or structural similarities with known plastic degrading enzymes using profile ED markup model, genome mining, functional domain analysis, and a custom local protein database. Our materials and method. First, what we did is to search databases, protein databases, protein databases that have reports of bacteria that are known to degrade plastic. After we searched them, we concentrated on 3 databases. First is the plastic database, that's plastic DB, the protein microbial biodegradation database, PMBD, and the protein enzyme database. These 3 databases were searched thoroughly to find if there's been a report of polyethylene degrading enzymes. After we found out this, we moved to profile ED markup model. Profile ED markup model is the statistical tool that search for conserved sequence patterns in proteins. Because, of course, we want to know if bacteria have enzymes that are known to degrade plastic. We have to search for their conserved motif. So we begin to look for means to search conserved motif. One of the probable means is to use profile ED markup model because this model search protein domains. Going forward, we did a thing. We decided to construct a database for ourselves. We used these reported proteins in the first materials and method to construct a database for ourselves. We wanted to check, we wanted to do something new, so we decided to construct a database ourselves. Afterwards, we did genome mining and comparative analysis of bacterial enzyme related to plastic degradation. How did we do this? We tested the database that we constructed with Bacillus species. Then again, we did another thing. We did a step further. We created a pipeline. This pipeline consisted of the working mechanism of profile ED markup model, another working mechanism from our data construction because we use Diamond to produce this database. Then we added something else. Just like I mentioned, one of the most important parts of in silico analysis to find out bacterial enzymes that degrade plastic is to search conserved motif because that's where the action takes place. Now we went ahead to incorporate a database, an online database that does that. That's protein family database, PFAM. We added PFAM. We incorporated PFAM into our pipeline. Now, our results and discussion. Like I earlier mentioned, we did a systematic review of plastic degrading enzyme in these 3 databases. In the protein enzyme database, we found only 1 bacteria reported to degrade polyethylene. Even though it had 2 strains, we found Rhodococcus robur. In plastic microorganism biodegradation database, we found 8 bacterial species that are reported to degrade polyethylene. They include Pseudomonas, Rhodococcus, Pinibacillus, Streptomyces, and Psychobacter strains. And finally, in the plastic DB, the third database that we set, we found 46 bacteria species reported to degrade polyethylene. However, just 5 of them were experimentally verified. This chart, out of the 3 databases, we found out that Pseudomonas species was the most represented genus. The 3 of them had a lot of Pseudomonas in them. Pseudomonas was mentioned over 10 times in the 3 databases. Now, we have this, the gold coloration represents our plastic microbial biodegradation database. The blue represented plastic enzyme database, while the green bar represents plastic database. We found out that some of the databases reported similar bacteria. For instance, Streptomyces was reported in PMBD and plastic DB. Also, Cyclobacter was reported in PMBD as well as plastic DB. Pseudomonas aeruginosa was also reported in both of them, likewise with the Rhodococcus rubber, and then Rhodococcus ubacus was reported in protein enzyme database and plastic DB. This overlap is telling us the strength of each of the strains that were reported. If a bacteria could be reported in more than one strain, it means that aside being experimentally verified, of course, we know that it's explaining the strength of such bacteria. Also, in the 3 database, we had 67 bacteria in total. 67 bacteria, 27 genus, and 6 enzymes. 6 enzymes were reported in total in the 3 databases. 6 of them were lacases, alkane monoxygenases, alkane hydroxylases, copper oxidases, Cyclobacter lacases, and rubber oxygenases. So this is a chart showing the 63 bacteria in total, the 27 genuses in total. When I say in total, I mean in the 3 databases that we set, and the 6 enzymes that are reported to degrade polyethylene in these databases. For the purpose of this study, we decided to remove redundancy among the databases, meaning that for entries, like I showed us in this slide, these entries, for instance, Streptomyces was found both in PMBD and in plastic DB. So for the purpose of these studies, we remove redundancies and do not report any bacteria twice. So we report each bacteria just once. And then for that, only 12 of the bacteria were used for the purpose of this study. We reported only 12 of them. After removing redundancy, like we removed those bacteria that appeared more than once and reported them as just a bacteria. Then, at the end of the day, we came down with just 12 bacteria for downstream analysis. When I say for downstream analysis, I mean that we use them to continue the research. Now, that's the first part of searching databases. We ended up with just 12 bacteria that were reported to degrade plastics after removing the redundancy, removing those ones that appeared more than once. Then our second objective is, our second item on materials and method is to use profile Eden Markov model. Like I earlier mentioned, I said that profile Eden Markov model is a statistical tool that detect conserved motifs in protein. This model uses AMA. AMA is also like a working mechanism of this model. Now, what we did, like I earlier mentioned, the last materials and method was this systematic review of databases. From the databases, we found that we came down with 12 experimentally verified bacteria. This 12 experimentally verified bacteria known to degrade polyethylene was used to further this study. Now, what we did was these 12 enzymes, we retrieved the FASTA files of these 12 enzymes from NCBI BLAST. After we retrieved the FASTA files, of course, there are 12 of them. For each one, we retrieved their FASTA file, and then we stored them together. Afterwards, we did CD-IT. CD-IT is a cluster data at high identity with transparency. This is just to also remove any gaps that are in the FASTA file, any duplicates, so that it gives us a clean FASTA file. After removing the redundancy, we carried out a multiple sequence alignment. We did a multiple sequence alignment because for profile ID Markov model, the blueprint of profile ID Markov model is multiple sequence alignment. Because multiple sequence alignment help us know the domains of proteins. Those ones that the functional part of proteins. And that's the blueprint of profile ID Markov model. That's the aim. Okay, let me put it that for if multiple sequence alignment is taken out, then we don't have profile ID Markov model. So it's important that we do a profile ID Markov model so that the tool will be able to function perfectly. Exactly. So the most important part of profile ID Markov model is the multiple sequence alignment. After we carried out the multiple sequence alignment, we decided to create a phylogenetic tree. Why did we create a phylogenetic tree? So that we can pick natural clusters. Natural clusters in the sense that in a phylogenetic tree, of course, these are clusters, yes. And then because we used our muscle to create these phylogenetic trees, all of them are natural clusters. Now we decided to pick not just 1, 2, but 3 clades because we wanted to test the strength of this profile ID Markov model. Now, how did we pick the clades in this phylogenetic tree? We used the bootstrap because the bootstrap is the measure of confidence, is the reliability. We use the bootstrap number because bootstrap number measures confidence. It measures reliability of these clades. That's why we use the bootstrap. And then so for group A, we used group A because they're really similar. We have a bootstrap number of 198. And then for group C as well, we have a bootstrap number of 188. And then for group B as well, we have the group bootstrap number of 98, 82, 58, and 92. Of this entire phylogenetic tree, group A, group B, and group C are the most confident of them. That's why we decided to pick these 3 clades. So what do we do afterwards? Each queries in this group, we retrieve their FASTA file from NCBI for the 3 of them, and then did another multiple sequence alignment. Because of course, like I mentioned, the blueprint of profile ID Markov model is multiple sequence alignment. So it's just like we're trying to help it to get the best result. Because in a case where multiple sequence alignment is not done for profile ID Markov model, we cannot trust the outcome. But in an instance where we carried out multiple sequence alignment, we can trust the outcome. So after retrieving the FASTA files and removing the redundancy, the model uses something called build, that's HMM build, to create like a database of its own. What does it do? We use mbuild to create a database from all the FASTA files that we created. Now, this mbuild pull all the FASTA files together and keep them as one. Now, it keeps them as a database somewhere. Then, we now use the model also has search. This search is, for instance, we used it to search against Uniprot KB because, of course, Uniprot KB is a database with a large protein body. Now, this search set our FASTA files. Our FASTA files from the 12 enzymes that we found, it searched it against uniprot KB. Why is this searching against uniprot KB? We need to know which protein has similar conserved sequences pattern with our own FASTA files. We did the FASTA file that we found there, we did search to make this FASTA file a database. Now, we made it a database so that we can search it against anything. For instance, for the purpose of this research, we searched the FASTA file against uniprot KB so that we know which protein has similar conserved sequence pattern with our own FASTA files. After doing that, we found out that the Markov model result came out that we had even though we had a robust detection despite low enthalpy. Now, this is where the significance of conserved motif come to play, because the model made us know that our entropy is low. In a case where we are just focusing on sequence homologue, maybe we will not have a result. But because the model does not focus only on sequence homologue, it focused on conserved domain, it made us know that, okay, yes, you have a low entropy, but some of the domains that we detected are similar to multicopper oxidases. Now, the M-search made us know that scanning our FASTA file against uniprot, it was consistent and retrieved numerous multicopper oxidases, including high confidence homologues from Microbacterium tuberculosis, Priscotella agglutinin, and multiple Rhodococcus species, all showing significant hits. Now, the model made us know that the repeated independence hits across all clades confirm multicopper oxidases as the most prominent enzyme linked to polyethylene degradation. Now, our profile hidden Markov model made us know that the cluster of our FASTA files, that is, all the FASTA files that we generated from the databases that we search, they are in tune with multicopper oxidases, meaning that yes, too, a larger percent of what we are doing is correct, because yes, multicopper oxidases have been reported to degrade plastics. That's for our profile hidden Markov model. And then we went further to create a database on our own. We used Diamond. Diamond is also a tool. We used Diamond to create a database on our own. Just like we did for profile hidden Markov model, that we used the 12 enzymes from the database that we searched. We used these same 12 enzymes to create a database. Now, this database was created by Diamond, and it has over 2.7 million sequences from a merged FASTA file. These merged FASTA files are from the 12 bacterial enzymes that are reported to degrade plastics from the databases. Also, this database that we constructed ourselves, we used the terminal. We were working on terminal, so we created it on the terminal. This database has about 800 million total protein residue, and this is typical of a medium-sized custom protein database. Now, what do we do after creating a database on our own? We have used these 12 enzymes that we found from the databases, from the protein databases, to create our own database. Now, we picked enzymes that have been reported to degrade polyethylene from databases. Then we created our own. We created our own ourselves. After creating this database, we decided to test it with Bacillus. We want to validate the database that we created. We want to be sure that, okay, what we created can detect enzymes that are able to degrade polyethylene from any bacteria at all. Now, we ran Bacillus species from, we generated Bacillus data sets from NCBI. We did that on our terminals and ran it against the database that we created. We found out that a total of 1,156 query proteins showed significant similarities to known plastic degrading enzymes, yielding 11,506 high-scoring segment pairs. What does this mean? It means that after we generated Bacillus data set from NCBI and ran it through our database, 1,156 query proteins were similar to our database proteins. Now, out of these 1,156 proteins, they had high-scoring segment pairs of 11,506. It means that when we picked our database and NCB and Bacillus, for every protein, there was a time that there was 11,506 of them had similar sequence at every point. That's why we have this. So this is just like an example. Now this is our known enzyme from the databases, those ones that we fetched. This is it. Now when we ran our Bacillus data set against our database, it showed us that just like this, there are times that there are 11,506 points where they were similar, where the query proteins were similar with the known proteins. But at the end of the day, we also found out that these 11,506, they fell between percentage identity of 41 to 60%, which is indicating moderate sequence similarity to known plastic degrading enzymes. This is also validation, showing that most of them are moderately similar to known plastic degrading enzymes. Then what we eventually did was to remove redundancy. How did we remove the redundancy? Just like I said, most of these 11,500, most of them were similar. After removing redundancy, we always use in bioinformatics, we always use cdit. Cdit is like a command to say, oh, check through this sequence, and anytime you find something that is similar, that is repeated more than once, remove them and give me just one of it because they are similar. At the end of the day, this 11,506 HSP became 34 unique enzymes. Then, these 34 unique enzymes were found. What we now did, because our database could not do KHO identification themselves, we picked these 34 enzymes and ran them through KEGG. And then we found that, in summary, they fell under three categories, which is xenobiotic degradation pathway, carbohydrate metabolism pathway, amino acid metabolism pathway. Of course, xenobiotic degradation pathway is explaining enzymes that are able to degrade complex polymers, just like plastics. Also, carbohydrate metabolism pathway is aligning with those enzymes that are able to act with carbon and hydrogen, as well as carbon, and carbon oxygen compounds. Of course, plastics, polyethylene fall under this grade because it has a carbon, and carbon hydrogen bond. Then finally, amino acid metabolism pathway. This group are able to metabolize the breakdown product of plastics. So our database also confirmed. So at the end of the day, we're able to conclude that our database can also be able to be able to tell us if there's a bacteria that has the potential to degrade polyethylene because of what we have made them with, because of the enzyme that we created this database with. And then finally, our pipeline. Like I earlier mentioned, our pipeline is like integration of all the methods that we did. First, it was profile reading Markov model. Second was the creation of a database with Diamond. But then our pipeline do not just look for sequence homolog, but it goes deep down to look for it looks at the conserved motifs, the conserved domains of enzymes. Now, the pipeline that we created involves using first the diamond that we created in our pipeline. And then in this pipeline, it also uses PFAM, database. That's protein family database. This protein family database is an online database. Our pipeline is created such that we incorporated PFAM in it so that it can be robust. Now, unlike other two methods, the profile marker model and the database that we constructed with Diamond, where we used the 12 enzymes that was generated from the systematic plastic review. For the purpose of this pipeline, because it's very robust, we used enzymes that have been reported to degrade polyethylene terephthalate, and polystyrene. Because this pipeline is robust, we did that to give it enough power, go beyond and above, check if this bacteria has any iota of enzymes that can degrade plastics. So in this pipeline, unlike our database that we constructed, where after we found the 34 unique enzymes, we started imputing it in KEGG to give us what are the functions, what are the pathway. But for this pipeline, we don't need to fetch unique enzyme and impute them on KEGG. This pipeline is all-encompassing. It does everything itself. We only need to impute. For instance, if we feel like, oh, I have a bacillus species, let me check if it could degrade plastic. This pipeline is going to check if it has the potential to degrade plastic. Afterwards, it check on KEGG if there's a possibility that, what is the function, what is the pathway. Afterwards, it does a gene ontology enrichment for that bacteria. So this pipeline is very robust, and that's the reason why we use 3 different experimentally verified plastics enzymes to build it. Now, these are the 3 different experimentally verified plastic enzymes that we use to build it. We use polyethylene terephthalate that has about 35 enzymes that has been reported to degrade it. We use polyethylene that has 5 experimentally verified. And then we also use 3 experimentally verified polystyrene enzymes to build this database, to make it robust enough to carry out the functions that we need. And then to remove the manual work of going from, oh, after you found an enzyme, check for me in KEGG. No, this pipeline is all-encompassing. It can do all of them itself. Now, after we created this pipeline, of course, we needed to validate it before we throw it open. We needed to be sure that this pipeline is enough to do what we want. Now, we tested it with rhodococcus rubber. Rhodococcus rubber has been experimentally verified and confirmed to degrade polyethylene. Now, so that we can validate if our pipeline is functional, we tested this pipeline with rhodococcus rubber. And then we found that after a total of 381,241 protein sequences were initially loaded, compared against our curated plastic degrading enzyme database using Diamond. Of course, I said that our pipeline has Diamond PFAM embedded, profile marker model embedded in it. Now, the first part was to use Diamond to check. After it found out 381, after filtering based on stringent threshold, the expected value of 1 times 10 by minus 5, an identity of greater than 30%. Because anything that is less than 25% is not homolog, they are not homolog, and anyone that is more than 95%, there's a lot of redundancy in it. Now, after that, we found that 523 high-scoring segment pairs were retained from the total 1,682 raw hits that were found. These homolog sequences were predominantly associated with hydrolysis and oxidoreductases, which is consistent with the enzymatic classes involved in polyethylene degradation. So how it works is that what we did was to impute rhodococcus rubber FASTA file into our database. What it does is that it does all the running, and at the end of the day, it comes out with a result template showing all the results. One of the results was that it gave us homolog sequence to hydrolysis and oxidoreductases, of course, which is consistent with polyethylene degradation. And then this is also generated from the pipeline. It generates it in itself. So the pipeline made us know that your sequence, the sequence we gave it fall between 32 and 44%, but most of them were reported under 32%, which is good enough for plastic degradation, for plastic degrading enzymes. Also, like I said, unlike the database that we created, we were the one that imputed them into KEG, but no, for the pipeline, it is all encompassing. It does it for itself. Now, the pipeline made us also know that the rhodococcus rhobarba you imputed into me, I found out that it is classified under benzoyl degradation, toluene degradation, and aromatic compound degradation. Of course, just as consistent with polyethylene degradation, the pipeline also let us know that most of them were under lacases, were classified under lacases. Some of them also were classified under monoxygenases as well as lipases, which is 100% consistent with the enzymes that are known to degrade polyethylene. Also, the pipeline does gene ontology term enrichment. Gene ontology is, it tells us what does a gene do, how does it do it, what is the importance of this gene. And now the pipeline classified rhodococcus rubber under biological processes, molecular functions, and cellular component. The biological processes involves, just like we see xenobiotic catabolic processes, where it's letting us know that these enzymes can act on complex non-natural compound, of course, which polyethylene fall under. Also for molecular functions, in making us know that the enzymes that we imputed in it are able to assimilate, break down product of plastics. And also for cellular component, most of them are, for instance, for this plasma membrane associated protein, there are proteins that are like on the surface, that are between inside the cell and the outside wall. So just like in plastic degradation, this could have been like, since they're outside, they are working on the surface of, just like residue, they're working on the surface like biofilms, just like biofilms, like creating, just like how during the degradation of polyethylene, the first thing that will happen is the production of biofilm from wear and tear of the surface. That's what this is explaining to us. And then also in the pipeline, like I said, we have PFAM embedded in it. We have PFAM and interproscan embedded in it. So the pipeline also gave us a report of PFAM or interproscan for rhodococcus, for polyethylene. And it makes us know that the FASTA files that are imputed into me, they are known for multi-copper oxidases with PFAM number of 0043. Also, they are found in hydrolysis fold domain, which are typical of plastic degrading enzymes. Also, they are found in catalase-related peroxidase domain, as well as lipase esterase domain, all of which are typical of degrading of enzymes that are known to degrade not just polyethylene, but plastics themselves. The conclusion of the study. This study demonstrates that integrating 3 different databases substantially expand the repertoire of bacteria associated with polyethylene degradation. This highlights the value of multi-database mining as reliance on a single source underestimates microbial plastic degradation. This explains that using more than one database as the basis of this research has helped us find a lot more enzymes. Because if we are stood with one enzyme, we might be stranded. We might not get the result. For instance, PASI had only one bacteria reported to degrade plastics. But because we took a step further and used more than one, we're able to get a robust number, and then we're able to use them for our study. We also concluded that the created non-redundant enzyme database minimized duplication and ensured reliable downstream analysis. Like throughout this study, I've been saying how we use CD-EAT. CD-EAT is like a statistical tool here that helps us to remove duplicates. And then because we are able to remove duplicates, it ensured reliable downstream analysis. The 34 unique enzymes with sequence similarity to known degradative classes, their functional assignment to KEGG pathway, such as xenobiotic degradation and carbohydrate metabolism, supports their possible role in polymer transformation. This finding suggests that polyethylene degrading capacity is phylogenetically widespread and non-restricted to a few mobile organisms, just like I've explained before. Overall, the validation of our pipeline using Rhodococcus rubber confirms its robustness in detecting and annotating putative plastic degrading enzyme homologue. This buttresses the fact that the results that our pipeline give us, of course, we saw how all of them are inclined to enzymes that are known to degrade plastics. So this supports the robustness in detecting and annotating putative plastic degrading enzymes homologue. Now, finally, the integration of KEGG, gene ontology, protein family, and blast-based analysis provided a multi-layer functional profile, suggesting that Rhodococcus rubber employs both oxidative and hydrolytic mechanisms during plastic degradation. And finally, I will say that our pipeline has demonstrated strength in detecting enzymes that are able to degrade plastics, not just polyethylene, but any plastic at all. These are the references of this study. Gracias por escucharme.
Agradecemos la participación de la maestra Aminat y pasamos a la sección de dudas, preguntas o comentarios que tengan los asistentes.
Thank you for the presentation, first of all.
Thank you.
My question is, are you planning to make in vitro assays with these results?
No.
Okay.
No, because to a very large extent, our pipeline has reduced lab workload. Our pipeline is able to detect we can give it a 80% validation. When it says that something is present, 80% of it is usually correct. So maybe out of 10, we might just validate one in the lab. So it has already reduced the part of the laboratory workload. So we might not really need to do the validation in the lab.
Thank you.
De nada.
Hi, Aminat. Thank you for your presentation. It was very good. I just got a question. And when you set the threshold for, well, when using diamond, was the threshold based on the consensus of domains or the whole sequence, protein sequence, I mean?
When I said the threshold,
yeah, that just, well, I understand that it was like a blast using diamond.
yeah, yes.
And you said the threshold, for example, I remember that you used 30% identity.
Yes.
So my question is, if that identity was based on the domain consensus sequence of each protein or your cluster proteins, or it was based on the whole proteins you got?
The domain consensus.
Oh, got it.
Yeah.
And any way to confirm those proteins are not coded by pseudogenes?
Any way to confirm what?
That if those proteins are coded by pseudogenes?
Well, the proteins were created from experimentally verified plastics. So already, they're already verified.
Okay, thank you.
De nada.
Gracias. Y hacemos entrega del reconocimiento a la maestra Aminat. Me permito leer. Instituto Politécnico Nacional, Centro de Biotecnología Genómica, otorgan el presente reconocimiento a la maestra en ciencias Aminat Olamide Shokunbi por su destacada participación como ponente en el Seminario Departamental del Laboratorio de Biotecnología Ambiental con el tema Análisis in silico de enzimas asociadas con la degradación de polietileno. Ciudad Reynosa, Tamaulipas, a 3 de noviembre del 2025. Atentamente, la técnica al servicio de la patria, doctora Amanda Alejandra Oliva Hernández, directora. Muchas gracias.